Hadirnya UT memastikan bahwa setiap warga bangsa berhak atas pendidikan tinggi di bawah kekuatan hukum tanpa adanya perbedaan. Lebih dari 2 juta lulusan berhasil menyelesaikan studi di Universitas Terbuka. Kami bangga sudah 4 dasawarsa UT menghadirkan pendidikan berkualitas tanpa batas.
UT selalu berfokus ke masa depan dengan tetap berpijak pada nilai-nilai dan budaya adiluhung Nusantara. Digitalisasi layanan pendidikan mempermudah masyarakat dan mahasiswa mendapatkan informasi mengenai UT. Selain tercetak, bahan ajar juga diberikan dalam bentuk digital guna memudahkan mobilitas mahasiswa. Dengan demikian, belajar bisa dilakukan di mana saja, kapan saja, dan bersama siapa saja. Digitalisasi pendidikan juga diformulasikan ke dalam loka pasar digital mata kuliah yang ditawarkan oleh Indonesia Cyber Education Institute atau ICE Institute. Beragam mata kuliah dari perguruan tinggi terkemuka di tanah air dan mancanegara ditawarkan secara online. Perpustakaan UT sebagai salah satu unit vital di perguruan tinggi menawarkan fasilitas digital yang memungkinkan mahasiswa, dosen, tenaga kependidikan, bahkan masyarakat umum untuk mengakses sumber pustaka secara lebih leluasa. Beragam layanan yang diberikan memastikan student engagement dapat dilaksanakan dengan better, cheaper, and faster. UT pada tahun ini mengedepankan internasionalisasi program sebagai upaya menyejajarkan UT dengan perguruan tinggi jarak jauh berkualitas di dunia. Selain menawarkan program internasional, akreditasi internasional pun dilakukan. International Trust yang tinggi akan meningkatkan kredibilitas UT dan lulusannya. Public trust terhadap UT ditandai dengan terus meningkatnya jumlah mahasiswa UT dari tahun ke tahun.
Semoga keluar dari sini makin berkah hidupnya, apa yang diinginkan tercapai, dan yang pasti ilmunya bisa bermanfaat buat semuanya.
Tidak saja jumlahnya yang meningkat, komposisi mahasiswa pada saat ini didominasi oleh generasi milenial dan Gen Z. Untuk menjaga kualitas akademik, UT melibatkan BAN PT, Lembaga Akreditasi Mandiri, dan International Accreditation Bodies. Bahkan UT satu-satunya perguruan tinggi jarak jauh di Asia yang secara sukarela dan berkala mengundang ICDE untuk mereview semua layanan di UT. Kita ingin memastikan bahwa layanan yang diberikan telah memenuhi kaidah-kaidah dan standar kualitas yang diberikan oleh perguruan tinggi jarak jauh terkemuka di dunia. Pada tahun ini, jumlah program studi yang ditawarkan berjumlah 47 yang dikelola 4 fakultas dan sekolah pasca sarjana. Kantor UT daerah yang tersebar di seluruh wilayah Indonesia dan di luar negeri memastikan layanan pendidikan dapat dinikmati oleh semua tanpa terkecuali, termasuk mereka yang tinggal di wilayah terpencil, terluar, dan tertinggal, serta di luar negeri.
Mungkin harus menunggu puluhan tahun atau ratusan tahun untuk membangun kampus di tempat ini. Puji Tuhan, dengan adanya UT, Saya bisa kuliah meskipun saya tinggal di tempat yang amat terpencil, seperti ini.
Tanah airku tidak kulupakan, kan terkenang selama hidupku. Biarpun saya pergi jauh, tidak akan hilang dari kalbu.
Meskipun saya TKI di Hong Kong, saya tetap bisa kuliah di UT. Setelah jadi sarjana, saya akan pulang dan menjadi entrepreneur yang tangguh.
Meskipun kaki ini jauh melangkah hingga kutub utara, saya tetap memilih belajar di kampus tercinta Universitas Terbuka.
UT menyalawi.
Dengan semangat 4 dasawarsa, UT hadirkan pendidikan berkualitas tanpa batas.
Engkau ku banggakan.
Welcome to the School of Distance Education at University Sains Malaysia. Before we explore the narrative of our school, let me provide a brief overview of University Sains Malaysia, USM. USM was established in 1969, marking its significance as the country's second university. USM offers courses ranging from natural science, applied science, medical and health science, pharmaceutical science to building science and technology, social science, humanities, and education. These are available at undergraduate and postgraduate levels to approximately 30,000 students at its 18 academic schools on the main campus in the island of Penang, 6 schools at the engineering campus, and 3 at the health campus. USM also has 26 dedicated research centers for a wide range of specializations which include archaeology, medicine and dentistry, molecular medicine, science and technology, Islamic development and management studies, and policy research and international studies. In 2024, University Sains Malaysia achieved remarkable global recognition, securing the 137th spot in the Quacquarelli Simons QS World University Rankings. This prestigious ranking underscores USM's excellence in academia, research, and global outreach. Further elevating its status, USM ranked 4th in the Times Higher Education Impact Rankings, showcasing its commitment to the United Nations Sustainable Development Goals, SDGs. These rankings highlight USM's impactful contributions to sustainability, education, and societal well-being. Through its diverse campuses and global engagements, USM continues to foster innovation and excellence, making significant strides in higher education and research on the global stage. Now, we turn to the journey of the School of Distance Education, SDE, one of the 18 academic schools based on the main campus. Established in 1971, we are proud to be Malaysia's first institution dedicated to open and distance learning, ODL, offering transformative educational opportunities to working adults across the nation and beyond. Our history is not just a timeline, it's a testament to our commitment to accessibility, innovation, and excellence in higher education. Our history begins in 1971 with the foundation of the Off-Campus Studies program, marking the inception of Malaysia's pioneering open and distance learning initiative. This initiative evolved into the Centre for Off-Campus Studies in 1984, found a new home with its dedicated building, and was later renamed the Centre for Distance Education in 1995. By 1999, our growth and success led to the establishment of the School of Distance Education. This evolution mirrors our adaptability and our ongoing commitment to meeting the educational needs of our students. Our vision and mission are deeply rooted in the belief that education should be accessible to all, transcending traditional barriers. Since our establishment in 1971, SDE has been the vanguard of multidisciplinary open and distance learning in Malaysia. We've led the charge from correspondence education to the latest online learning technologies, setting the standard for ODL both nationally and regionally. SDE is not just an institution, it's a pioneer in the field of open and distance learning in Malaysia. From the outset, we've embraced every evolution of educational technology, transitioning from correspondence courses in the 1970s to the advanced online learning platforms of today. Our dedication to instructional design and technology has made us a model for other ODL institutions, contributing significantly to the national ODL blueprint. Our journey is marked by a commitment to innovation, research, and the development of teaching methodologies that resonate with our diverse and dynamic student body. The backbone of SDE is our dedicated team, consisting of 72 academic and 38 administrative staff members. SDE offers 4 undergraduate programs, 3 master's programs, and 1 PhD program. SDE is led by our esteemed top management, including the Dean of the School, Associate Professor Dr. Zulnaidi Yaqub. He is assisted by 3 Deputy Deans, Dr. Hazrul Abdul Hamid, who oversees academic, career, and international affairs. Associate Professor Dr. C. Kok Fong is our Deputy Dean for Research, Innovation, and Industry Community Engagement. Dr. Chua Han Kuan oversees Technology and Learning Support, and Mr. Mohammed Rashid Abdul Rajab serves as our Deputy Registrar. We have 12 program chairpersons in areas including biology, physics, chemistry, mathematics, political science, economics, anthropology, sociology, geography, history, literature, management, and university courses. Together, we are committed to upholding our mission, driving the success of our undergraduate and postgraduate programs. We offer a broad spectrum of undergraduate programs in ODL mode, including Bachelor of Arts in History, Bachelor of Social Science with majors in Political Science, Economics, and Anthropology Sociology, and Bachelor of Management covering Finance, Organization Management and Strategic, and Marketing, alongside Bachelor of Sciences in Biology and Mathematics. These programs are crafted to empower students with the knowledge and skills needed for success in their respective fields. Our approach to education combines web conferencing, lecture, lab sessions, and self-instructional modules, accommodating the needs of over 5,187 current students. This model supports active learning and engagement, demonstrating our commitment to delivering a comprehensive and effective educational experience. With more than 20,000 alumni, our impact extends far beyond the classroom, contributing to a global community of professionals and leaders. Our postgraduate programs are as diverse as they are in-depth, offering Master of Arts, Master of Sciences, Master of Social Sciences, and Doctor of Philosophy degrees in fields ranging from distance education, educational technology, TESOL, geography, history, literature, management, biology, chemistry, physics, mathematics, environmental science, economics to political science and anthropology sociology. Our research-driven approach, open to both local and international students, ensures a vibrant academic environment that fosters innovation and scholarly excellence. The diversity of our student body, including 128 international and 85 local students, enriches the learning experience, fostering a global perspective within our community. Engagement beyond the classroom is vital. Our postgraduate students partake in seminars, webinars, conferences, and community engagement projects, enriching their educational journey and fostering a sense of community and collaboration. These activities are pivotal in cultivating a dynamic academic atmosphere and preparing students for the challenges and opportunities of their future careers. In 2023, SDE's commitment to excellence was evident through our significant achievements, securing 53 research grants, publishing 116 indexed articles, including 40 in Q1 and Q2 journals, establishing 15 international collaborations, engaging in 13 industrial linkages, and launching 24 community projects and outreach activities. Additionally, our focus on microcredentials and MOOCs underscores our dedication to innovative and accessible education. As we look ahead, the School of Distance Education continues its commitment to offering high-quality, flexible learning opportunities. We are proud of our legacy as pioneers in open and distance learning and excited about the future. Thank you for joining us today. Let us venture into an exciting journey of learning, research, and innovation together.
Welcome everyone. Good morning from Indonesia. It is a pleasure to have you all here in this our third webinar series from the Biology Study Program, Faculty of Science and Technology, Universitas Terbuka, which is titled the Biology Webinar, Bridging Biology Across Borders. This is the third series of the webinar, which is in association with University of Sains Malaysia, Apex, Pusat Pengajian Pendidikan Jarak Jauh, Universitas Terbuka. First, to honorable speaker for today, Ibu Dr. Amirah binti Hurzaid from the Biology School of Distance Education, University of Science Malaysia. Selamat pagi, Ibu. Semoga kabar baik dan sehat selalu ya.
Yeah, Alhamdulillah.
Yeah,
Thank you for having me today.
yeah. Thank you. Thank you, Ibu Dr. Amirah. It is our privilege and our pleasure that you will becoming the speaker for this international webinar series, and we are very much looking forward to it. And also to Honorable Ibu Dr. Subekti Nurmawati, the Dean of the Faculty of Science and Technology, and also the speaker for today's webinar. Selamat pagi, Ibu Nurma. Semoga
Selamat pagi, Mas Fauzi.
Selamat pagi, Bu. Kabar baik dan sehat selalu, insya Allah. Thank you very much to all the participants, fellow lecturers, and also the students who joined this webinar. And yeah, I think we can now moving forward into the main lectures for today's webinar. But before that, please allow me to read the CV of both of the speakers. I think to the IT team, I would like to ask for the CV of Ibu Amirah. Okay, Ibu Dr. Amirah binti Hurzaid. Ibu Amirah take the PhD in Marine Oceanography from National Taiwan University with the dissertation titled Molecular Phylogeny and Diversity of Paneidae in Southeast Asian waters. And the scope of her research interest is very broad and specific as well. From molecular taxonomy, zoology, marine invertebrates, population genetics, phylogeography, biodiversity, integrated approach in taxonomy and environmental biology. So currently, Ibu Dr. Amirah is a senior lecturer in Biological Science Department, School of Distance Education, University of Science Malaysia from October 2020 until now. Beforehand, she is a research assistant in the Center for Marine and Coastal Studies in the USM, and then research officer in the School of Distance Education USM. Also, the teacher in the Seberang Perai District Education Office North. And there are a lots of academic contribution achievements from Ibu Dr. Amirah as a principal investigators. Perhaps I will read some highlights of it. Her project including the enhancing sea-based farming through innovative air-ice technology, and then elucidating the cryptic diversity and potential commercial marine shrimp species, and then also population genetics, diversity and taxonomy of Mears, Paneopsis, Hardwicki, and Sculptilis, and etc. So I think that is the excellent portfolio of Ibu Dr. Amirah binti Hurzaid. But before Ibu Amirah deliver the lecture, I get the message from the committee that I think it's the best that we take the preliminary photo session together first. So I invite all of the participants here to turn on the camera so that our team can document it, please. I think it's still only 10% of the 78 participants turn on the camera, but I invite, we will wait for several seconds. I think, please, to all of you, turn on the camera because we will have a documentation. Or maybe we will take again by the end of this webinar. But we will have several versions of it, so perhaps to make a good comparison in between the webinar. Okay, I think now it is already good. Okay, please, for the team, we can document this all the participants. I can take this as well. Okay, 1, 2, and 3. And then the second slide, third slide, and the fourth slide. So now we have almost 80 participants, and maybe we can wave our hands to the camera for the documentation. And then, okay, it's good. Thank you. Okay, now without further ado, please, Ibu Dr. Amira Hurzaid, we invite you to deliver the lectures.
Okay, thank you very much, Mrs. Fauzi, for the interesting introduction about me. So I am now trying to share my screen. Hold on, I need to move this cursor. Okay, can you see my slide? And my voice is okay?
Yes, the voice and the slide is very good.
Okay. Okay, Assalamualaikum and good morning to everyone, okay, to honorable guest speaker, another guest speaker, the docent Dr. Subekti Nurmawati, and all lecturers from both university and students from both universities. So first thing first, thank you very much for having me and to share my work entitled Integrative Taxonomy Reveals Hidden Diversity in Southeast Asia, PNID. My name is Dr. Amira Hurzaid from University Sains Malaysia. So basically what I'm sharing today is my project, started my PhD project actually, started from my PhD and still continue until now. Okay, so what is biodiversity? So biodiversity means a variety of life on Earth, so variety of living organisms that live on Earth. And under biodiversity, there are several terms, which are species diversity, genetic diversity, and ecosystem diversity. And for species diversity, it means that different organisms, different living organisms like plants, animals, fungi, and even microorganisms, okay, there are falls under species diversity. And then next is genetic diversity. Okay, genetic diversity meaning a variety of gene within species that helps population to adapt to changes. So changes like, for example, like climate change or even disease. So this is genetic diversity. And then the third one is ecosystem diversity. So ecosystem diversity, by definition, is the range of habitats and ecosystem. So whatever habitat and ecosystem, we call it as ecosystems diversity. For example, like coral reefs, they have their own ecosystem diversity. Mangrove, seagrass, bats in marine environments. Okay, so under biodiversity, again, I repeat, there are three terms, okay, species, either species diversity, either genetic diversity, or even ecosystem diversity. So now you know what does it mean by biodiversity. So next, what does it mean by hidden diversity? So hidden diversity is also known as cryptic diversity. Okay, and cryptic diversity refers to the existence of distinct species yang kita nampak clear, okay, physically, that are not easily distinguished by appearance alone. Or in other words, it means that there are the species is morphologically similar but genetically different. When we look based on their physical appearance, they look similar, but actually their genetic are different. So this is what it means by cryptic diversity, is a hidden diversity. And hidden diversity is not not uncommon. Okay, it has been found in many animal groups, including marine organism. Like here is nudibranch, taken from Sorensen et al. So based on morphology, they look similar actually, and then further analysis found actually there are cryptic diversity, cryptic species of nudibranch and amphibian. And this one is from Rowley et al. 2015, based on the frogs. Okay, and this hidden diversity can only be revealed through molecular tools like DNA barcoding. So why finding hidden diversity is important? Okay, first thing first, we need to understand the true biodiversity. Okay, because species, they look similar, but they are genetically different. So by discovering them, we can give us like a more accurate picture of the life diversity. Okay, next we want to improve species identification, or in other words, we want to like prevent misidentification. Okay, why? Accurate identification is important because it is critical in conservation, okay, and for like marine organism in fisheries and even in ecological research. Next, why we need to find hidden diversity is we want to protect endangered species because some of these hidden species, they are maybe rare or threatened. So without recognizing them, they might go extinct, unnoticed. Maybe they will extinct. Next is by finding the hidden diversity, we can guide the conservation efforts. So once we can identify them, so then we can provide specific management system, so that we can protect their resources, right to their population or even ecosystem. And last but not least is we want to reveal evolutionary relationships because hidden diversity actually it helps scientists to trace evolutionary history, and we can understand how species adapt to different environments. So these are the reasons that I can find why we need to find the hidden diversity. Okay, now let's move to my topic of research. Okay, I focus on Pinaidae. Pinaidae reference like 1815. Pinaidae is actually a family of marine shrimp, so occur in marine area, not freshwater. And Pinaidae, there are economically or commercially important crustacean, and their distribution is worldwide. Okay, they can be found in shallow, inshore tropical, subtropical waters, and the highest species diversity is recorded in the Indo-West Pacific region, which include our countries, Malaysia, Thailand, Indonesia. And this family also known as primitive group of decapod crustacean. So what does it mean by primitive? Means that it exists like long time ago, even before dinosaur exists. Okay, so this is why you call it as primitive. And this is example of udang minyak. In Malaysia, we call it as udang minyak, or the scientific name is Miaspinapsis hardwicki. And I'm very sure all of you knows will definitely find this family, in the market, in the landing sites. So because they are commercial. And these are examples of Pinaidae species. Okay, this one is, of course, all of you know, I think, black tiger prawn, widely cultured, or in the scientific name is Pinaeus monodon. We also have like white leg prawn. Okay, this is the most cultured species, Lithopenaeus venemai, originated from Brazil, but now we culture extensively in the Southeast Asia. And kuruma prawn or Marsupials japonicus. There are many other examples, but these are the most common one that we can find. Okay, and this figure shows you the general characteristic of Pinaidae. So basically, Pinaidae, the family look like this. They have like rostrum, yang tajam ni, we call it as rostrum, well developed. Their cervical short, this is carapace, their head region, and this is their abdominal segment. Their telson, sharply pointed. This is their pleopod. We call it as swimming legs. And this is preopods. Preopod is walking leg. And this is third maxilliped, antennuloflagella, and then kita panggil misai dia lah. Kalau orang Melayu, Malaysia kita panggil misai, udang. Okay. So for Pinaidae, the first character, if you want to like differentiate between different families, Pinaidae, the observed character is that their first segment overlap with the second segment. Okay, for udang galah, or freshwater prawn, macrobrachial, the second segment will overlap with the first and third. So for Pinaidae, first they always overlap each other. Started from first. Okay, so this is basically the general characteristic of Pinaidae. Okay, so how to identify the species within the Pinaidae? So this is very interesting because the identification of the species is based on their shape of genitalia, the sex organ. Okay, and this species specific. So this sex organ, we call it a male petasma. For male, we call it as petasma, and this is located here in between the first pair of leg. Okay, and for female, we call it as telecum, located here. Okay, and they are under their thorax, their carapace. And this character, the shape is species specific, meaning that different species has different shape. And however, this character is not useful in juvenile specimen. Why? Because their genital organ is not fully developed yet. Okay, so it's only useful for the adult specimen or adults. So this is another example. Okay, example like identification of Penaidae based on the genus. So if you can see here, my left figure is telicle, the male genital area, and this is petasma. Sorry, I changed. Actually, this is petasma, this is telicle. Okay, and this is for Penaeus. And even for each species, they has different shape. Okay, this Parapinaceae, this I just show you the major one. Okay, so how many species of Penaidae exist? In 1990, Dahl et al., they described 200 species under 17 genera. Later in 1997, 7 years after that, the Paris, Farfante, and Kensley, they found 217 species under 26 genera. So why this genera is like add more from 17? Because they split the 17 and then they found many more characters. So they conclude that there are 26 genera within 217 species. And then in 2011, okay, Sakai and Shinomiya, they proposed another genera within the Parapinaceae. Okay, I will not explain it later because it's too like molecular. And then finally now, family Penaidae include more than 240 species under 33 genera. The whole world. Okay, to identify Penaidae is very challenging because they have varied larva stages and sexes. Okay, like this is their example of their larva stage. And then the morphological traits used may be differ across classification system, and the traits may not be non-homologous or lack phylogenetic value. Non-homologous means that it looks similar but it's evolved independently. And because of this, results are sometimes ambiguous or incorrect. So that's why we need molecular data to help for accurate identification. So some advantages of molecular data, it is useful for deciphering intraspecific and interspecific variation, which this information is valuable for species identification. And molecular data like DNA, RNA, okay, they can also eliminate morphological convergence by ecological adaptation, which is very useful for phylogenetic studies. So for species identification, usually scientists will choose cytochrome, oxidase subunit 1 or CO1 gene. So why the CO1 gene is suitable for species identification? Okay, because clear difference between species but is similar within a species, meaning that they have enough variation to differentiate between species. Okay, and CO1 gene is a part of mitochondrial DNA. This is maternal inheritance, and it is easy to extract and amplifies well, and it can work with a universal primers. And CO1 also has a large reference database, which make the species comparison easier. So this figure shows you example of the mitochondrial gene, okay, in Microbrachium rosenbergi. Okay, so CO1 located here. This is mtDNA. Like CO1, usually scientists, they will use like partial CO1, but in Penaidae, because it's non-ambiguous, so we have two, Fulmer and Palombi. Okay, next, now you know diversity, okay, you know Penaidae and molecular. So what is an integrated approach in taxonomy? So integrated approach meaning the combination of multiple criteria to delimit species, meaning we don't use only one criteria. For example, traditional identification, they will use one criteria. For example, morphology, okay, taxonomists, they will prefer to use morphology. Okay, maybe some advanced people, scientists, they prefer to use molecular data. Okay, however, for integrated approach, it means that we combine two or more criteria to define a species. For example, we can combine comparative morphology and biogeography together, and we define a species. Or we combine morphology and ecology based on the area. Okay. Or molecular data and behavior, or comparative morphology and molecular data. So meaning that integrative approach in taxonomy, we combine multiple criteria to define or delimit species. Okay, so now we move to Southeast Asia. Okay, Southeast Asia located within Indo-Pacific region. So this is the map of Southeast Asia. And it lies at the heart of the coral triangle. Coral triangle is a high diversity of coral. And Southeast Asia is known as a center of biodiversity for both marine and terrestrial taxa. And previous studies has documented the presence of cryptid diversity even on marine animals like fish, okay, oyster, and even the terrestrial. Daratan pun ada banyak cryptid diversity. Yet, however, study on shrimp is still limited. That's why I conducted the study on shrimp. So the objectives of our project now is to first to explore species diversity in Southeast Asian waters using integrated approach in taxonomy. And second, we want to resolve the taxonomy of certain species complex. Species complex meaning the potential species that we found in the first objective. Okay, this is the sampling areas. This one conducted in 2020, but now we expand. Okay, we have that data more in Malaysia. And in total, we have 32. We collected samples from 32 locations covering countries as Malaysia, okay, Vietnam. We went to Vietnam at this area, and then Indonesia, specifically in the Aceh part, okay, and then Thailand here, and even Taiwan. Taiwan is not considered as Southeast Asia, but some people say Southeast Asia. So we just collected here to compare. And this is my student, my master student, Afiq, is conducted his sampling in Terengganu this time. So basically, we just go to this landing site. Okay, we wait for the boat to come out to the shore, and then we go and collect the specimen. Okay, so when we collect the specimen, first thing first, we want to identify their morphology. So basically, we use these two guidebooks. Okay, when we get the specimen, we identify to the species level first based on their morphology, and follow with the molecular experiment and analysis. However, if we collected juvenile and unknown species, what we do is we identify to the genus level. We go to following the molecular experiment and analysis. After that, the result will be re-evaluated after the Cowan gene, meaning we will check which clade it belongs to, which species it belongs to based on the clade. Okay, and then we check back the morphology. For the molecular data collection, okay, we cut the Force-Player pot, tissue cutting, and then follow with DNA extraction, either using DNA extraction machine or kit, or even CTEP protocol, traditional method. And then proceed to PCR and then sequencing. So the results here, and then after that we will analyze their DNA based on the Cowan gene. So for to delimit species, okay, these are the two analyses that we use. First one is ABGD, which based on distance, and we use input sequence file, the DNA file, and then BPTP web server. This is tree-based. The input is the tree file. And then we compare. And right now, I think this software is no longer available, but they replaced with ASAP. Okay, actually, basically, it's the same. The principle is the same with ABGD. So if we conducted these two analyses and the results is congruent, meaning the identification is correct, or it shows the robust support of the OTUs. OTUs mean operational taxonomic unit or potential species. Okay. However, if incongruent, if the result is not similar, we need to add additional criteria. For example, we can conduct nuclear DNA analysis, okay, morphology, and geographical distribution. We check back to confirm whether there are true species or not. Okay, so based on our finding, so this is the result, okay, part of the result. Okay, the input sequence, we use like 371 individuals, okay, and based on morphology, we identify 71 species of penyait existed in the Southeast Asia. However, after we conducted this analysis, we find, okay, ABGD 91, BPTP 96, then we compare. There are several steps to check, and then final 94. So it shows that there is underestimated biodiversity on Penaidae in Southeast Asia. So from 71, now we have 94. And the potential cryptid species occur among these species. Okay, however, the one that I crossed is a sequence downloaded from GenBank. So meaning that we cannot check the morphology, so we cross out. Okay, and then we found that monodon 1, 2, 3, 4, 5, 6. So 6 potential cryptic species existed within this morpho species. And then I will produce the result, 3 results. Okay, so basically after we got the result, we compare. Like this is if there is congruent results, okay, we confirm. Okay, this species based on multiple criteria confirm that there are morpho species. Okay, but okay, if there is incongruent, then we need to compare. Okay, and then make a decision. Okay, so hidden diversity we found in Pineus monodon complex. Okay, the tiger prawn, and then we found in Pineus coromondelica. Okay, and then Cationeopiasis and Myospinopsis. And the findings we published in journal Zoologica Scripta, published in 2020. In this paper, we also discuss about the phylogeny, okay, and evolutionary history. But diversity we also included here. And here we just during this publication we just mention how many potential cryptid species existed without further morphological evaluation. And then after that, from that, okay, we focus on each species complex. So first we focus on Pineus monodon or tiger prawn. Okay, there are 4 clades or 4 potential species. However, for we call it like P1, SP1. Okay, first, second, third, and fourth we couldn't identify. We still did not find the morphological difference. Okay, even geographical barrier we couldn't find. And then this one, okay, Pineus CF monodon P1 SP1 collected from Indonesia, Aceh. Okay, so we collected in 2020, we collected 8 individuals at the west coast of Aceh in Lambeso and Meolaboh. Okay, and then we cannot like compare because 8 specimen is limited. And then I revisit the area. Okay, I think I went there in 2021. Went there again to Jambu Masi, Aceh Jaya, it's still Aceh province, and collected many more specimens with the help of the local farmers and my friends there. So we collected many specimens. Okay, I think the total is 47 of Pineus CF monodon, and we observed the morphology. When we got many samples, then we can make a comparison. For example, the Pineus CF monodon, okay, the rostrum is longer. Okay, for tiger prawn, shorter. Okay, and then the bending, okay, CF monodon doesn't have bending, clear bending, and monodon tiger prawn has like a distinct bend on their body. And the finding of this journal we published in Q3, Journal of Crustacean Biology, published in 2021. So right now we confirm, okay, the Pineus CF monodon from Aceh actually is a Pineus simplex. Okay, and this species presents only off the western coast of Aceh, and it is considered as endemic, so we must protect them. And this species has high potential for the economy because it's related to tiger prawn, so of course it has a high potential for economy. And then next, we focus on Cationeopiasis complex. Okay, there are 2 morpho species, okay, K. incisa and K. cornuta. So K. incisa, okay, so for K, this species complex, based on their male genital organ, we didn't find any difference. Okay, they look similar. However, for their telecum, the female genital organ, okay, there are difference between these morpho species. So K. incisa, they look like this. Okay, and then K. cornuta, it look like this. There are difference. However, among the potential species, okay, we are like here we have example. We have 1, 2, 3, 4, 5. 5 we couldn't find the morphology difference. So next we focus on K. incisa complex based on publication. Our finding published in 2020, we found 2 inferred species. However, during that time, morphological comparison between these species cannot be done because K. incisa is from East China Sea language, East China Sea lineage, and low sample. And then okay, this is my student Farhana, went back to the area. Okay, we collected many more specimens. Alhamdulillah, we were lucky because we able to collect the additional specimen. And then we conducted both morphology and molecular analysis. And finally, we confirmed the first distribution record of K. incisa in Malaysia. Before this, it only existed in China. Okay, and now we confirm that this species also existed in Malaysia. And for K. cornuta complex, so this is interesting finding. So first one, we found 5 inferred species, okay, cryptic species. Okay, this is the true one. Okay, if we want to know whether which clade is the true cornuta, true species, we need to compare with their type locality. Type locality meaning the first time the species described. Let's say it's from Japan, you need to find collaborator in Japan, ask them for their morphological examination or even the DNA analysis, better DNA result, so you can compare with your result. And in 2020, we think that the distribution of this potential cryptic species is based on the geographic factors. This one is collected in Aceh as well, in Indonesia, Aceh from Strait of Malacca. IO is Indian Ocean, and this is from Malaysia. So Malaysia from Strait of Malacca, and this one is from Sabah, Sarawak. From Sarawak, Sarawak, Malaysia, Bornean part. However, because we have low number of sample, we cannot confirm. Just like from their external morphology, looks like whether they have stripe or not. That's it. And recently, early this year, we confirmed that one of the clade is K. maxillipido. So it is no longer K. cornuta. And for the other two species complex, there are Hatwicki and Scaptilis. There are 3 inferred species. And this is my student, Afiq, conducted this study. So he collected many, around 100 individuals, 108 individuals, and we conducted further morphological identification and confirmed them as one species. Okay. And then we conclude that they distribute, okay, they separated based on geographical barriers, but actually they are the same species. And the finding is under review. Okay, same with Scaptilis. Okay, we add more individuals. This is M. Scaptilis or rainbow shrimp. We found there are separated based on geographic barrier, but still there are same species. So here is the summary of finding. Okay, so in total we have seek, okay, monodon, tiger prawn, 4 OTUs, soft only 2. Okay, incisa, okay, done. Okay, we already resolved. Cornuta, still only soft 2 OTUs, 3 still waiting. Okay, Hatwicki and Scaptilis done. Coromandalica still ongoing. Okay. And this is my future plan, pending resolution. If anyone here want to collaborate with me, then you are mostly welcome. Because first, cornuta is very interesting. So now we successfully, this one is true cornuta. Okay, this one, the second clade is the K. maxillipido now. So 1, 2. The first one we collected only single individual here from Aceh. Collected here only 1 individual, AC 65. And then 4 individuals from this side. Okay, tadi this the other side, Indian Ocean side, and here is from Strait of Malacca side. We also collected here from Malaysian side, Strait of Malacca, but there are different species based on this. So still ongoing. Okay, actually it's not ongoing. We start here. We don't have like a collaborator to help us collect more. Okay, hopefully, if anyone here wants to collaborate, you are most welcome. So conclusion and call to action. So why this research matters? Because it fills a critical knowledge gap in hidden diversity of commercial marine shrimp in Indo-West Pacific, particularly Southeast Asia. And this research, I hope, will open new pathway for future research and application. Okay, thank you very much for your attention. If you have further question, you can ask later for me. Thank you very much. Back to Mrs. Fauzi.
Thank you very much. Terima kasih banyak Ibu Dr. Amira for the excellent presentation. Would like to confirm that the Q&A session will be delivered after Ibu Nurma giving the lecture. So we invite all the participants. If you have question, you can put it in the chat box, and our committee will compile it for us. And then also you can ask directly to Ibu Dr. Amira, but please hold a moment because our Q&A session will be available after Ibu Dr. Subekti Nurmawati deliver the lecture. But yeah, once again, thank you very much. I think we can give a round of applause using this Zoom tools. I think, yeah, let me check. Okay, React, we can give a very great round of applause for the excellent presentation from Ibu Dr. Amira. Ibu Dr. Amira explaining comprehensively about her research project regarding this integrated approach in taxonomy, especially combining different methods to investigate novel organisms, or to identify the similarity and the phylogenetically the Paneidae organisms using molecular data, comparative morphology, and based on the different sample in the biogeography around the Southeast Asia region. So now I think we can move forward into the lecture from Ibu Dr. Subekti Nurmawati. Ibu Dr. Subekti Nurmawati, before they deliver the lecture, I'd like to ask the IT team to display the CV. Okay, this is the CV of Ibu Nurma. So Ibu Nurma finished her PhD study in Biology Study Program in IPB University, and the work of Ibu Nurma is from 1991 until now. Currently, Ibu Nurma is the Dean of the Faculty of Science and Technology in Universitas Terbuka. She also had experience in the human resource development manager, also the vice dean of the faculty, also the head of the biology study program. Several publication of Ibu Nurma is related to this Munun organism, Munun longipetalum, or from the Anonaceae family. This is the publication in 2019. A new species from Sumatra, Indonesia. Also about Munun kalimantanins in the Anonaceae family, a new species from Kalimantan, Borneo, Indonesia. Also, the title of the third paper is this excluding the species of Munun fragrans Anonaceae from the flora of China. And then the last one is about the environmental research about the waste management analysis of South Tangerang City, an alternative review of solving waste management problems in urban areas. So Ibu Nurma, please, you can deliver your lectures regarding this diversity and distribution species of Munun in Borneo, Indonesia.
Okay, thank you, Mas Fauzi. Ya, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Best wishes to all of us. And I would like to present about the Munun is one of the genus of Anonaceae. At the first speaker talked about crustaceae, and now I would like to talk about plant, one of the genus of Anonaceae. Let me share my PowerPoint. Okay, can you see?
Yes, this is clear, Ibu Nurma.
Okay. 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 I'll start the presentation. The title is The Diversity and Distribution of Munun Species Anonaceae in Borneo, Indonesia. The research is based on the morphological character, just focus on morphological. What is Munun? Munun is one of the genus of Anonaceae that is the family that well known is some example Anona muricata, Anona squamosa, Annona ceremonia, and also Annona reticulata. We know that this fruit is as an edible fruit. One of the species of the Munun genus is Munun longifolium. This is synonym with Polyhadria longifolia. That is, as we know, as ornamental plant that we can easily find in in road. Okay, and I would like to about the short history of Munun. The family included family of Anionaceae, subfamily Malmeoidae, and the tribe Muliusae, the genus Munun. The genus Munun, it was first established by Miquel in 1965. However, the Miquel circumscription was heterogeneous. Some taxonomies, for instance, Bentham Hooker and also Sinclair, Housden and Kessler, treated Munun as a section of Polyalthia. Polyalthia was established by Bloom. Polyalthia, we know that Polyalthia is the largest genus in Indonesia. There are 155 species, according to Renner and Catrow. The genus was polyphyletic. It means that the species origin from many ancestors. An attempt to disentangle Polyalthia, morphological and also molecular analysis have been done. As a result, some species removed to other genera, such as Trivalvaria, Disepalum, Green Wayodendron, Venerifia, and also Marsipopetalum. And also from this research, two new genus. There are Masia and also Huberanta. Based on the phylogenetic study of Polyalthia done by Su et al. On 2012, Munun raised as a phylogenus. And one species, Munun lateriflorum, as the type genus. And this new genus, including Polyalthia section Munun, and also Enicosanthum, Woodilantha, and also Cleistopetalum. This genus we call Munun sensulato or Munun SL. It's mean Munun in broad sense. Two new species also published by Normawati, Munun longipetalum, and also Munun kalimantanense. And what is the uses of Munun? Some examples of the uses of Munun species. Munun longifolium as a cytotoxic compound against cancer cell, hypertension, and Munun cupulare and Munun membranifolium as an antimicrobial compound, and also Munun lateriflorum as fungicide compound. Distribution of Munun in the world. The Munun species widespread in Sri Lanka, Malay Peninsula, Thailand, Philippines, Sumatra, Java, Bali, Sulawesi, Papua, Solomon Island, till Australia. And also the most diversity is found in Borneo. The objective of the research is to describe the diversity of Munun species in Borneo, to describe the distribution of Munun species in Borneo, and also to compile the distribution map of Munun species in Borneo. The materials and method. Materials collection from Borneo, the herbarium, dried materials, stored in herbarium, Leiden in Sandakan in Singapore, Wana research, the total of herbarium examined is 363 collection collected from 1965 to 2017. Type specimen from some herbaria also investigate in the world and some literatures. Fresh materials also observed in botanical garden, Bogor and also Singapore, and conservation forests of Wana Reserve in East Kalimantan. Taxonomic revision methodology uses mark stats in 1992 and 120 vegetative and generative characters, qualitative and quantitative characters were examined. The botanical terminology used in this study, followed by Harris and Harris, for general structures of plant, used then for typical flowers of Annonaceae, Panseton and Cook Norman, for typical fruit and seed of Annonaceae. The general characters of monuns, actually the monuns has the habitus S3, and also the unique characters. The species of munun has longitudinal furrows, mitrid abaxially, and secondary fins, Eucamptodromus, where the minis to the leaves margin. Munun species has 6 petals in 2 horns of 3. The single ovule by the name of munun, we call to this genus because it has single ovule. And then the current insertion of secondary fins, secondary fin types is scalariform, and the fruit size along more than 1.5, and also wide more than 1 centimeter. The seed with longitudinal groove like this, and also soft endosperm lamelliform. From the investigation of the species based on the morphological characters, I can found the 23 species from Borneo, widespread in Sabah, Sarawak, Brunei, North Kalimantan, East Kalimantan, South Kalimantan, West Kalimantan, and also Central Kalimantan. There are 23 species. And this is the distribution map of munun in Borneo. The most species we can found in East Kalimantan, Sabah, the most is Sabah Sarawak, and the secondly in East Kalimantan, North Kalimantan, West Kalimantan, and Brunei. Sorry. Okay, this is the distribution of munun in Borneo. In Sabah, we can find 17 species. In Sarawak, 12. In Brunei, we can find 7 species. In North Kalimantan, 9 species. East Kalimantan, 16. South Kalimantan, 12. West Kalimantan, 8, and also Central Kalimantan, 7 species. The distribution area of munun in Borneo, the most species we can find in the distribution is Munun lateriflorum. This species can find in all of the area in Borneo, and also Munun longifolium can found in all of the area. The morphological characters, the munun species can be distinguished based on some characters such as the shape, texture, and size of leaf lamina, inflorescence positions, type, shape and size of petals, shape and size of fruit. The longest leaf is monon grandifolium. The thickest leaf is monon coriaceum. The thinnest leaves in monon membranifolium. The most densely hairy is monon erianthoides. Leaf apex and base leaves variation. Leaf apex variation acuminate can find in monon lateriflorum, abruptly acuminate in monon anomalum, rounded apex in monon coriaceum, cuspidate in monon sclerophyllum. Based on the leaf base variation, we can find the cordate shape found in monon grandifolium, obtuse base in monon hypogaeum, rounded base in monon lateriflorum, and cuneate in monon sclerophyllum. This is the position of inflorescence, and we can find axillary on supra-axillary or ramiflorus. This is the example of species of monon, monon anomalum, lateriflorum, longifolium, and also monon sclerophyllum. We can find in inflorescence type as a many flowers in fascicle in a group, and we can the type of inflorescence is solitary or few flowers. We can found in monon sympatalum, monon glabrum, monon membranifolium, and also monon paradoxum. The ramiflorus position sometimes we can found in monon sclerophyllum, and cauliflorus also found at this position in monon hypogaeum. According to the stamen and pistil observation, the stamen, the number of stamen is numerous, spirally arranged, tightly packed at the torus, and all glabrous or hairy. The two types of the stamen and pistil. The pistil numerous, the surface is glabrous, or hairy. This example of species is monon hookerianum, monon borneense, monon membranifolium, and also monon longifolium. The apex, the shape of apex is capitate and glabrous. Glabrous, it means no hairy. Monon batranterum, monon lateriflorum, and also monon congregatum. The surface apex hairy. The area between the two thecas is hairy, found in monon amiscocarpum, monon erianthoides, and monon grandiflorum. Carpels, we can find the stylus and ovary. The example in the species of monon anomalum, monon batantrellum, and also monon lateriflorum. The ovary found in hairy, and the stylus found in glabrous. In the three species, monon longifolium, ovary glabrous, and stylus hairy, and monon congregatum, both of ovary and also stylus in hairy, and monon sympatalum, both ovary and the ovary and also stylus in hairy. The variation of fruits, we found that in some species, the shape is cylindrical, glabrous in monon lateriflorum, globus glabrous in monon longifolium, deltoid glabrous found in monon amiscocarpum, fusiform, short apicule found in monon borneense, fusiform with long apicule found in monon hypogaeum, the shape deltoid, densely hairy, found in Munun cupulare, subglobus, hairy, found in Munun santopetalum, and subglobus with short-pointed apicule found in Munun hookerianum. The varieties of Munun malateriflorum, Burlak in 1901 divided Polyalthia lateriflora into 5 varieties, included Calak variety, Macrophylla, Elongata, Siamensis, and Typica. But Sinclair in 1955 did not divide the species into some varieties. In this research, I've divided the Monon lateriflorum into 3 varieties. First, variety of lateriflorum, variety of Calak, and variety Macrophylla with the differences of character. These varieties, under taxa under the species of Monon lateriflorum, can divided into a macrophyllum, Calak, and also lateriflorum based on the characters of young leaves, flowers, and also fruit variation. We can see that the young leaves of Monon lateriflorum, there are 3 types with the color of light green, almost red, and also dark green. And the flowers, we can see that the 3 varieties, we can see 3 types, the color. There are red, dark yellow, and also light yellow. Based on the fruit, we can find typically the fruit is the same types, but the differences is the start of the fruit. And in this research, I propose one new combination that is Munun spatulatum. Originally, Munun spatulatum is from Polyalthia spatulata. The basionym of this species is Guatria spatulata. Why I remove this species from Polyalthia to Munun, because it has Munun's characters. For instance, leaf-base symmetry, the current, the insertion of secondary venation to midrib, Eucamptodromus, and also the seed. It has 1 seed. In research also, research identification key. So we can identify the species by key I propose. And the conclusion of the research, the diversity of Munun in Borneo, there are 23 species, 7 new records, 2 varieties, 1 species proposed as a new combination. 15 species have been recognized as published by Turner in 2011, and then 2015 and 28. 7 species as new record is Munun congregatum, Munun glabrum, grandiflorium, macranthum, membranifolium, passifilum, sclerophyllum. And the distribution of Munun, the highest is in Sabah and the lowest is in Central Kalimantan. 2 varieties proposed is for Calak and also Macrophyllum. The one new combination is Munun spatulatum normawati Komnov. That's all. Thank you very much.
Thank you very much, Ibu Nurma, for the excellent presentation, and we can give the big round of applause digital in the Zoom feature here. So thank you once again, Ibu Nurma, for the great presentation regarding the diversity and distribution of the Munun species from the Anonaceae family in Borneo, Indonesia, focusing on this comparative morphology methods to identify the diversity of the organisms. So now we have come into the Q&A session. I would like to invite all the participants. If you have question, you can send me through the chat Zoom or through my WhatsApp message. I can give or I can deliver to the speaker as well. So feel free to ask. Also, by asking, you can also unmute your microphone to speak directly to the speakers. We are open for that as well. If anyone, by raising the hand. Okay. Perhaps I can start asking question based on the message that I get. Perhaps to Ibu Dr. Amira. There is a question here that is sent to me asking about the regarding the sample of the shrimp that you obtained from Aceh. Is it solely based on because of the species is endemic in Aceh, or other consideration why you choose Aceh as one of the sample from Indonesia? Is it closely geographically close to Malaysia, or yeah?
Okay, very interesting question. Okay, first thing first, because I have collaborator here, a collaborator there in Aceh, so that's why we just go. That time, our strategy is we just go to all places that we have collaborators and collect the samples. And then during that time, based on morphology, we couldn't identify them yet because we collected juvenile and very limited shrimp from Aceh. So the main reason is because I have collaborators there. They help me do the sampling. I think Aceh is quite big area, so we went there by 4-wheel drive. We spent like 2 weeks there, and then we collect the sample first. And then after that, I went back to Taiwan. Okay, did the morphology check and do the molecular test, and then we found, oh, Aceh is very interesting. That's why I say it's hidden diversity. And then after that, we want to go back, but we have, of course, no money. So that's why the sample, I mean, the identification stops there. The research stopped there only for monodon because it's not monodon, simplex, the endemic species, because actually the villagers, the fishermen, they already noticed there is a new species there, but because they have lack of knowledge based on morphology and taxonomy, and if I'm not mistaken, one of the, I forgot the name of the guy. He is the fisheries government officers in Aceh. So he told me that they think there is new species, but they have no like knowledge to conduct the experiment. Okay, so they publish it. Actually, they already published, but because they published is wrongly. Okay, I mean there is like some, for publication you need to follow certain rules. Okay, and then they give the name. This is sayang lah. So they give the name Pinayas Aceh. Actually, I intended to use the word Pinayas Aceh, and then because the name is invalid, considered invalid, and then the publication is not accepted. It's published, but the name is not invalid. So we need to come out with another name. At first, we want to choose Milaboh and Aceh, Milaboh and Lambeso, But then because the species is not specific to either of that area, so we need to find other name. So we cannot use Aceh, we cannot use Milaboh, we cannot use Lambeso. So finally, we use simplex based on it lacks of bending pattern. So that's the story actually behind that.
Thank you, Bu Amira. And perhaps it's also a bit of perhaps my lack of understanding, but I just want to make sure that the focus on your research is for the shrimp that is being cultured in the coastal areas. Is it naturally living there, or it has been farmed by some local farmers or local authorities? Because I saw that this is like you take a sample of the shrimp. All of them are in the coastal areas of your location of the sample.
Okay, so I collected all my shrimp is a wild specimen, it's not cultured.
Okay.
So that's why I need collaborators for each countries to bring me there.
Okay.
So it's all wild. Yeah.
Thank you. And also there is this follow-up question from my colleagues. So if Can we say that based on the diversity of analysis, diversity and phylogeny analysis, can we say that this shrimp that is biogeographically uh, geographically near, are they even closely similar, or do you find some interesting patterns uh, of this closely related or similarity of the species and the geographic location of it?
Okay, it's interesting. It depend on the species. Some species, even they are biogeographically related, closely related, they look different. Okay,
Okay.
other species, even they closely, I mean the area, okay, biogeographically close, they look similar. So it depends on the shrimp itself. That's why. That's why
Also,
we.
also based on the sequencing that you say, also they are even the pattern is like sometimes it's
Ah.
closely similar. Okay, yeah, yeah.
Sometimes, okay, actually I didn't show in my presentation. For others, some species like Scaptilis, I don't know. At Indonesia, we have a rainbow shrimp, the one that I mentioned in the last slide. I mean they look different based on the biogeography. But for monodon, okay, remember I mentioned there are four clades, okay, for potential species. One is belong to Aceh, one is the true one. So between these two, okay, this is a problem now because we take the breed, okay, the true breeder from the wild, okay, but actually from Strait of Malacca, okay, this here at this area, and then from South China Sea, they are mixed together and form two different lineage. Okay, I mean both lineage have uh, same sample from the same area, like both lineage. Let's say lineage one, they have Strait of Malacca and South China Sea, and then the other lineage they have Strait of Malacca and South China Sea, and they are morphologically similar. So it's very difficult to like make a conclusion based on monodon. So yeah, sayang lah that we don't
Yeah.
know what to do. Yeah, maybe we need expertise from others. Maybe you have Indonesia, you have more knowledge, then we can share the
Yeah,
potential.
sure. Yeah, we can discuss more about it. But
Ah, yes.
UT in UT, I think we don't have the like specific experts in this field. Oh, actually, Ibu Ernik, Ibu Nurma, can we say that Ibu Ernik? Yeah, but maybe Ibu Ernik is focused on the farming aspect or agriculture aspect, isn't it, Bu Nurma? Maybe you can,
Yes, right. Bu Ernik
Bu Ernik. Yeah, yeah.
get in crustacea also.
Ustaz, yes, okay, yeah, we can, we can discuss more about it. Thank you very much, Ibu Dr. Amira. And I will now read the question itu Ibu Nurma. There's a chat in the chat Zoom here from Hamdani. I think I'm not sure whether sorry, Ibu Hamdani or Bapak Hamdani. I'm not sure. And whether it's from UT or USM or our partners. So our Hamdani says, considering that mono species are found in the various forest types, such as primary lowland forests and peat swamp forests, how do different habitat conditions influence the morphological diversity of mono species in Borneo?
Okay, thank you, Mas Hamdani, for your question. I can explain that Indonesia, especially, some they have habitat in primary forest, primary lowland forest, and very rare I found in peat swamp. So in this research, the lowest species is found in Sintra Kalimantan with the habitat peat swamp, and some one species that widespread in all the area is one species is Munum latriflorum. When the when it found in peat swamp, the leaves is more smaller, smaller than compared with other area. There are many some characters that different that found from peat swamp and also primary lowland forest, like this.
Thank you, Ibu Nurma. So that is the answer to Pak Hamdani's Mas Hamdani's question. And then from Sebastian Wong, Mr. Sebastian Wong from the USM, as to Bu Nurma, it would seem that perhaps Kalimantan may be considered somewhat less botanically explored and under-collected compared to Sabah and Sarawak. Based on Ibu Nurma's experience, would you expect a potentially higher rate of novel species to be discovered in Kalimantan?
Yes, of course. I think there are still many species that cannot be still have not been found yet in Kalimantan. And I believe that there are many herbarium stored in many herbaria unidentified, included in Malaysia, Sabah, Sarawak, and also other area in Malaysia Peninsular. I think there are many species that has not been found and identified.
Yeah. Yes. Thank you, Ibu Nurma. Uh, I'd like to invite the Zoom participant again. If you want to ask directly by voice. Okay, I will read through my messages that I get here. Okay. Uh, perhaps now this is to Ibu. I think it's for Ibu Dr. Amira. There's one fellow student here ask, how can the discovery of the cryptic or novel species in Southeast Asia Penide affect shrimp fisheries management and sustainability efforts in the region, or whether if it's even relevant to that?
How the hidden diversity will affect. Okay,
Yeah.
like I mentioned in my earlier slide, okay, hidden diversity is like if we found, for example, I choose example from Modern from Aceh because that one is very closely related with tiger prawn. So we know that that species is endemic to that area only. So in terms of fisheries management, okay, further research need because I just like my research is like the first step. So we know that now we have endemic species. Next is other people, like they maybe have knowledge on genetic diversity, like population genetics. So they need to collect sample and then they need to check the diversity, the level of, there are certain term like you need to check nucleotide diversity, okay, haplotype diversity to make sure that this species can how to say can maintain the environment or not, or if let's say something happen, okay, let's say climate change happen, so maybe will impact their distribution. Okay, so for in terms of fisheries, I mean it's not only me, anyone works on diversity, hidden diversity. So our like work is to find the species, and then we based on publication, okay, or maybe like talk like this, we give like awareness. Okay, we found something, okay, so and then fisheries need to take action. Okay, and then so another person will work on that, okay? Or maybe me myself, okay, I will like, usually for Malaysian, I will work directly with fisheries department. And each region, I think Indonesia, you have your own. Maybe I tell my collaborator, and then my collaborator will find a way, okay, to like give awareness and then discuss with the fisheries agencies in Indonesia. So that's example. And for Malaysia, so far, um, just based on the publication, we publish and then we tell them and then we share the information like talk like this, okay, and then maybe further discussion directly with the fisheries management agencies, okay, whether like what is the next step, what we need to do, okay, maybe we like implement like rules, say do not overfishing, okay, do not collect more than needed, okay, if you collect juvenile, release it back. That's like, I mean, the further step. As for my role right now, we just need to give awareness and then mention to the public. Okay, that's it. I hope my answers clear.
Yes, very clear. Yeah, thank you, Dr. Amira. And I think that, yeah, now next question from Bapak Ari from UT Jakarta, asking about this cytochrome oxidase subunit 1 gene analysis is kind of like a gold standard in for the sequencing, especially for in relation to uh, uh, identifying this perhaps novel organism. But whether it is have a prospect or relevant to be implemented, it's in kind of like a biodiversity monitoring by local authorities or in aquaculture industries.
Yes, yes. For Kowan is a standard marker. Like I mentioned in my earlier slide, okay, Kowan is like a universal because they has like several advantages. Like they have universal, so everyone use it, so we can compare. Usually we put our sequence, we submit to the gene bank. So from gene bank we can download, we can compare from other countries, and then we can identify. So that's why I think Kowan is better, but it depends on the organism. For example, I also have knowledge work on amphibians, particularly frog during my master's degree. So for frogs, we use 16S. Okay, for plant, we use IT. I think Dr. Norma knows better. So I think plant, they use different gene. I mean, each organism, they have like their marker gene, but Kowan is the well-used among all organism.
Yes, thank you. But it's a bit now, sorry, one more follow-up question from him. So during this DNA extraction, what particular tissue that we usually can get from the shrimp to extract the DNA and to sequence it for the CO1 gene?
Okay, I always choose the second last leg. Okay, they have like swimming leg, and then they have I
Oh,
mean
leg, yeah, the leg.
leg
Okay,
ya,
yeah,
kaki
yeah,
dia tu, okay, kaki yang keempat, kedua last.
yeah.
Yang ambil ada because usually based on my experience when we collect, sometimes they lost the last leg, sometimes they lost the first pair of leg. So
Okay.
I usually use the reason why I choose the second last because if you want to take picture, so we can standardize. Okay, so usually so the answer is we always collected the legs, the legs.
Oh, okay. I
So
thought it
it's
would be in the abdomen. No, okay. The leg because it might be a bit tiny for the leg. It's not the most uh the biggest area. Okay, yeah, but it's very interesting. Yeah.
so we don't destroy the specimen. So it's
Okay.
like
Yeah, 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 yeah. Thank you. Thank you, Ibu Dokter Amira. I'll come back again to Ibu Nurma now. There is a question here from Fauzan in UT Bogor. I'll check this here to my message. Is there any challenges in identifying this monon species when we are in the field directly? And if Ibu Nurma can mention the practical solutions if we try to identify it directly in the field for this monon species?
Okay, it's good idea to collect in the field if you want, but we need to study the all species have been published before, all species. So if we want to study in the field directly, sometimes we face difficulty, and then we have to share the specimen to herbarium and ask for the expertise. And in taxonomies, I think not all taxonomies can know all of the plants, but they have specific area, specific species, specific genus who can overcome to the species. So I think if you have difficulties to identify directly in the field, I think you can ask and accompany by the expertise.
Okay. Thank you, Ibu Nurma. Now, question again from Mas Fauzan. Oh, he's from UT Jakarta actually. So, what is the main challenges in distinguishing this monon species taxonomy? Because Ibu Nurma mentioned sometimes we need to go to the herbarium to double check or to do some more even look into more data or samples. But the thing is that the herbarium specimen sometimes that date backs to maybe half a century or 40 years of age of the sample. Perhaps Bu Nurma can give an insight if there any if it become a challenge or not really a challenge because it's preserved.
Most challenges that
Okay.
when we explore in the field is when we found the vegetative specimen. No flowers, no fruit. It's very difficult because the leaf characters sometimes similar one to each other. And in herbarium, there are many type specimen. Type specimen where the publisher, the first publisher, based on that specimen as a type, so we can study the type specimen to confirm what we are identified from the species. Yes.
Thank you. Maybe this one last one. Let me choose again.
This is one question from Al-Mahira.
Oh, okay. Is it in the? Yeah, sorry. Yeah, from I think Mbak Al-Mahira from UT. She'd like to ask Ibu Nurma about the identification of Anonense specimen in your study. Was there a particular reference book or herbarium-based collection that Ibu Nurma used? Additionally, are there any published records or comprehensive checklist available that document the distribution of Anonense species across Indonesia?
Okay. Someone who interested in Indonesia, there are many books, references, uses to identify, for example, in my presentation. Okay. In my presentation, we can use Van Housden. Van Housden, 1992. Van Housden published the book, very comprehensive. Van Housden compare one by one based on the genus, the typical flowers in Anonense. So we can compare and define which one the A genus, B genus, something like that. And also the next one is Van Zetten and Cook Norman, based on the typical of fruit. So some fruit in Indonesia, we can differentiate by that book easily. And then another two books, publication of some from the name is I'll check again. From okay. From Paul Kessler. Yeah, Paul Kessler is one of the expertise who interested in Indonesia. The book can browse in some herbaria or in digital, so we can find easily the digital publication of Paul Kessler, and also no, that's right. And also Turner, Turner is one of the expertise in Indonesia who interested in Munun also. Okay, that's all.
Yeah, thank you, Ibu. And I think now it's the last question in the Zoom chat that I will read from Satish from Distance Education, the University of Sains Malaysia. To Ibu Nurma, it seems there is more to go for discovering unidentified botanical species. How Ibu Nurma discovering that there is a place or field with unidentified species, maybe in Indonesia or in other country?
Okay, I think there is more to go for it. Okay, the specimen I identified in some herbaria, it's with the identity in the specimen, not in the specimen. Very clear who found, who collected, who collector, and when the place the number of collection, and also the habitat, the ecology of the specimen. So I can confirm that specimen is right from the collector.
Okay. Yeah, thank you, Ibu Nurma. If anyone want to have a feedback to both of our excellent speakers today, you can you can unmute your microphone. Okay, then I think that will do it. We are coming to the end of the session. Once again, thank you very much, Ibu Dr. Amira, Ibu Dr. Subekti Nurmawati, for the time giving the great lectures in this webinar, third series of this webinar. And yeah, but before we close this session, we would like to present both of our speakers with a token of appreciation. I think first to Ibu Dr. Amira. To the team, please. Yeah, this is the certificate of appreciation to Ibu Dr. Amira. And please, Ibu Dr. Amira, you can maybe deliver your closing statement for our forums here.
Okay, first of all, thank you very much for having me again. So hopefully we can continue this collaboration for other projects. Yeah. And thank you. I will like especially thanks Dr. Nurma, okay, and then Mrs. Hikmah and Dr. Novi, okay, for helping, and also our MC for today, Fauzi Zuhairi. So if anything, you can contact me. And if you have further question regarding my project, or we want to have like future collaboration. Harapnya ukhuwah kita ni bersambungan lah sampai bila-bila ya.
Ya, terima kasih sekali Ibu Dr. Amira. Maybe one quick question, I wonder if you have a long weekend in Malaysia, because in Indonesia we have this in Monday and Tuesday we are in holiday of this Waisak, the Buddhism, I think it's Buddhism, yeah, Buddhism holiday. In Malaysia, do you have it?
So in Malaysia, in Indonesia, you have 3 holidays for Wesak.
Yeah, from Monday and Tuesday we are not. Yeah, it's yeah, we will not be in office in Monday and Tuesday. What about in Malaysia?
Oh, so interesting to know. In Malaysia, we also holiday, but only on Monday. So we have like long weekend from today until only one day for for Wesak.
Our government then being gracious for extending. But yeah, for sure, we'll look forward for the collaboration. I think Ibu Nurma will deliver it as well. But yeah, now the second token of appreciation, our team will give it to Ibu Nurma. And Ibu Nurma, please, for the closing statement.
Okay, thank you very much, Mas Fauzi, and also Biologist team for this webinar. I really appreciate for your coordination and also collaboration between USM and also FST. Hopefully, the collaboration can develop rapidly. Yes. Okay. Thank you.
Thank you, Ibu Nurma. And now we turn on all the web camera. We have already turned it on in the beginning of the webinar. Now we will document it again. Please, we will wait for several seconds for all the participants to open the web camera. Come on, come on, come on. We know that this is the long holiday, but please turn on the camera for several seconds. Then we can, continue with our activities. Okay, I think that will do it now. Our team will document it. 1, 2, 3. We can say waving goodbye to the camera. The second slide now, I will document it. The third, fourth, fifth, and sixth. So now we have more than 130 participants coming in here. Thank you very much. Hopefully we will learn together more about these great topics in biology, about diversity. Ibu Dr. Amira mentioned the novel technology and methods for discovering cryptic species in crustacea organisms, and Ibu Nurma deliver lectures about this diversity in monon species. Great and relevant for all of us as a biologists and scientists. And yeah, thank you very much for attending. Best wishes and good health. Thank you very much for all the committee, Ibu Hikmah, Bu Nailah, Bu Novi, Mas Ari. And yeah, we will see you again in the next opportunity. Bye-bye. Thank you.
Bye.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Dr. Amira.
Terima
Dan
kasih.
teman-teman, ya, dan
Untuk
teman-teman
datang ke
semua.
Penang, maklumkan
Ya,
saya.
ya.
Aku nanti
Two
deh.
months, 2 months ago, I went to Malaysia.
You did when?
Yeah, graduation of my sons.
Oh, I forgot. Yeah, yes, so yeah, I didn't.
Okay, just one day, just one day.
Oh, just one day. That's why, because
Yeah,
I remember you told me that you want to have like a
okay.
collaboration with Dr. Zul, right? So I'm still waiting. So what happened? So maybe something come out. Ya Allah.
Come in the morning and back to Indonesia at night.
Oh, cepat sungguh. Yalah, mungkin kita boleh teruskan nanti kan
Ya,
bincang.
terima kasih.
Terima kasih ya. Jumpa lagi ya semua. Fauzi, Kamala.
Ya,
Ya, Dokter Amira. Thank you, Bu Norma.
ya,
Thank
makasih
you,
Mbak
Bu
Ima,
Novi, Mas
Mas
Fauzi.
Fahmi, Mas Fauzi.
Ya.